Good morning, everyone. With this short video, I'm gonna give you an overview on this study entitled Application of Animal Rennet and Plant Coagulant in Italian Caciotta Cheesemaking. Comparison of sensory, biochemical, and rheological parameters. In modern cheesemaking, the use of plant coagulant is increasing year by year due to ethical and religious choices. One of these plant coagulants is obtained by the cardon plant. This extract contains cardosine, an important protease able to coagulate the milk. With this study, we wanted to test this kind of extract in a local production to see if it's a really good alternative to the animal rennet. To try to answer to this question, we conducted the experimentation in a small cheese factory in the northern region of Trentino Alto Adige in Italy, where starting from raw milk, we produced two different caciotta cheese, one coagulated with the animal rennet, and another series prepared with the cardon extract. Both samples were tested for chemical composition, color, sensory analysis, microstructure, and texture profile analysis at time 0 and after 10, 30, and 60 days. Results, with the exception of color, where no difference has been detected, highlight several differences between the two samples. Starting from the sensory analysis, it appeared that the cardon caciotta was more complex, adding some herbal and floral notes to the sensory profile. Also, the microstructure was very different, with the cardon caciotta presenting a more compact structure. This was also reflected in the texture profile, where the cardon caciotta appears as harder and more gummy. Also, the chemical composition highlights some difference, in particular, a lower water retention and a lower presence of soluble proteins was noted in the cardon caciotta. This allows us to conclude that this plant coagulant had a lower proteolytic activity. To conclude, we noted some difference between the two samples, but it was interesting to see that the caciotta prepared with the vegetable coagulant seemed to have a higher degree of complexity related to the sensory profile. It was also interesting to see that both samples were welcomed by the panel and that the bitterness usually associated with this kind of plant coagulant was not detected in this study. For this reason, we believe that cardon extract could be a valid alternative to animal rennet, and in this specific case, so for a local producer, it can represent a new opportunity to open new markets. Thank you for the attention.